Click for IB Biology. Here we're looking at the cell cycle in terms of the chromosomes and the stages of mitosis: prophase, metaphase, anaphase, and telophase. There's a blank PDF of this presentation down in the description that you can annotate with your own notes. There's a quick 10-question quiz with answers. Sometimes there are channel links. There will be an AI prompt which will allow you to do self-assessment with your favorite chatbot. So let's take a look at the phases of mitosis within the context of the cell cycle. So let's just recall, we've already looked at the idea of G1, S, and G2 as being subsections of the interphase. When we observe the cell, the nucleus would be in place like this. And this raises a very interesting point, which is, why is it that the prokaryotic cells do not engage in mitosis? And there are two reasons for this. The first reason why prokaryotes do what's called binary fission, the reason they do that and not mitosis is, firstly, they only have one chromosome to replicate, and that is called the nucleoid. Whereas, of course, multicellular and all the eukaryotic cells have multiple chromosomes. The second reason for the necessity of prophase, metaphase, anaphase, and telophase is that eukaryotic cells have a nuclear membrane. And with the nuclear membrane, this means that even with DNA replication, the replicates, the pairs of chromatids, are inside the nucleus. And so we need to get rid of the nuclear envelope in order that we can separate to form our two daughter cells. So as we go into more detail, we've already discussed how in G1 we have gene expression and growth, specialization of the cell, and therefore specialization of the function as genes are switched on to express and form that cell. As the cell matures, it prepares itself for cell division through the replication of its chromosomes in the S phase. And each of the chromosomes present will end up as two copies called pairs of chromatids. So here are chromatids, and the pairs are held together by a structure called the centromere. So that happens during the DNA phase, and so now we've got the double set of chromosomes necessary for the two cells, the two daughter cells that we're going to form. But we need to replicate our organs, our organelles rather, and that takes place during G2, the mitochondria, chloroplasts. Many of the organelles replicate themselves, and then we enter the phases of mitosis itself. The first one is the very necessary breakdown of the cell membrane, of the nuclear membrane, which occurs in the prophase when the chromosomes appear. So in this image here, we can actually see the chromosomes. They're still randomly assorted around, and this is characteristic of the prophase. The metaphase, the second stage, will see the pairs of chromatids aligned across the equator of the cell, and so it would look very much like this. And so the equator of the cell is in this axis here, and the preparation is for the anaphase. Now the anaphase is when the two chromatids are pulled apart, and you can see this in this diagram here where the copies of the two chromosomes, one going this way and one going that way, for each of the two new daughter cells. And that has to happen for each of the chromosomes that the cell has. And characteristic of the telophase is the formation and the bundling of two sets of chromosomes at opposite ends of the cell, where the number of chromosomes here is equal to the number of chromosomes there. The number of chromatids has been maintained, and so now the cell is ready to separate the cytoplasm, which of course is the process of cytokinesis. So what we're going to do is we're going to look at the details of prophase, metaphase, anaphase, and telophase so that we're aware of the necessity of each of the stages there. And so the link is coming up here to chromosome condensation, a late interphase process, prophase process, to the channel link, and of course to the playlist. Please do subscribe. It's encouraging.